All right, welcome to WSQ number 13. Today we're going to be talking in chapter 4 about section 3 on cell division. And really two objectives we have for today. One, we're going to learn what events take place during the 3 states or stages of the cell cycle. So we're going to learn what they are. And secondly, we're going to learn how the structure of DNA actually helps account for the way in which DNA gets copied. So we're going to talk about DNA transcription or replication. So a couple really important things to deal with. Let's start today with just starting out by explaining what the cell cycle is. The cell cycle is a regular sequence of growth and division that cells undergo. This is something that happens if you've grown, as we all have. Your growth is actually due to the fact that your cells are constantly growing and dividing. And so as they grow and divide, you actually grow in size as well. We also need to understand that the process includes 3 different parts or steps. Okay, it's going to include what's known as interphase, it's going to include mitosis, and it's going to include cytokinesis. And so one of the first things we're going to do is we're going to describe how these 3 things actually work. What are we talking about when we talk about interphase, mitosis, and cytokinesis? So that's our first step. Now, I also want to remind us that this is a cycle, okay? That the cell cycle, it's cyclical. So what's going to happen is the cells are going to go through this process. They're going to grow, right? Then they're going to go to a process that's going to be like our interphase process. Then they're going to have mitosis, and then they're going to finish up with cytokinesis. And it will then start again. So one cell will divide into 2 cells, which will divide into 4 cells. So what you hopefully will start to see, and then so on and so forth. That's what we're talking about with the cell cycle. The first stage of the cell cycle is known as interphase. Interphase is the period before cell division. So this is basically 3 things happen during this process. Ultimately, what's happening, the cell is growing, it's making copies of its DNA, and it's preparing to divide into 2 cells. So growing, what we're talking about when we say that the cell is growing, what we mean by this is this is where it actually grows to its full size. So we're going to have a fully mature cell, right? And it's going to have all of its different organelles. So all the different organelles that are necessary for the cell to function properly happen during this particular example. Okay, they'll have ribosomes, they'll produce endoplasmic reticulum, all the structures that are necessary. Then you have a stage known as DNA replication. This is where the DNA gets copied. Remember, DNA is vitally important because this has the blueprints, okay, for the cell, for the organism. What makes us who we are, what makes a banana what it is, what makes a giraffe what he is, what makes a rose bush what it is the DNA. And one important thing, if it's going to reproduce, if we have one cell and we want that cell to divide and make 2 cells, it's really important that both cells get a full copy, a complete copy of the DNA. Now think about this. If they didn't, then each time the cells would reproduce, you would lose information. And the loss of information in an organism is not good. Okay, and so it's really important that we have that DNA. The third thing that happens in interphase is this is the preparation for cell division. So you start to see changes, drastic changes that happen in interphase. This is primarily, there's some cells that are produced, different structures produced in the cell that are only used for during this process of division. Like the centrioles we'll talk about, and the fibers, the spindles, those kinds of things will happen here. So stage 1, the first stage is interphase, and we'll talk about it more in a minute. The second stage is mitosis. During mitosis, this is when the cell's nucleus will actually divide in 2. So the big key here is the division of the nucleus into 2 brand new nuclei. So if I have my cell here and inside I have my nucleus, what's going to happen is this nucleus is actually going to divide. So what we end up having at the end of this process are 2 new nuclei. That's what happens in mitosis. And DNA, you get a complete copy of DNA. That's really tiny to write DNA in each of the nuclei. So each of them are going to be distributed. Now, ultimately, when this splits and divides, what you get are 2 daughter cells, okay, complete with their own nucleus, their own DNA, perfectly functional. That's what's going to happen in mitosis. We'll talk about that in more detail in a second. Now, I need to show you this cell cycle real quickly. We're going to talk about the third step in a moment, but I want to go over very quickly what's going on in each of these stages specifically. So let's talk about interphase first. Interphase, remember, this is the stage at which you have growth. Okay, so it grows to mature size. 2, it's making a copy of its DNA. And 3, it's preparing to divide into 2 cells. So we can see this, right? It grows, copies its DNA, and prepares to divide into 2 new cells. That's what happens in the interphase. And what we see is you see these little things here that show up in the cell during the interphase are called the centrioles. We'll also notice that the chromatin, which was in the nucleus, which at one point was very kind of free-formed and opened up, okay, and spread out, is now starting to get really dense and connect together, and it's going to ultimately become the chromosomes. So we've got the centrioles, we have the chromatin starting to change during interphase. Now mitosis can be divided up into literally 4 different stages. Okay, and you need to know these 4 stages. The first stage of mitosis is what we call prophase. And prophase is this 2A stage in mitosis in which a couple things are starting to happen. You have the formation of these little things right here called spindle fibers, which form between the centrioles. And these centrioles are basically these little structures which are going to end up pulling the cell apart. Inside the nucleus, you'll notice that the chromatin, which was free-formed over here, is now starting to form into chromatids or into chromosomes. Okay, so we start to see the condensing of chromosomes in the cell, and the spindle fibers and the centrioles move to the opposite sides of the cell. The last stage of this, what happens is this little nuclear envelope, okay, starts to break down. So the envelope that has kept certain things out of the nucleus and kept the nucleus from the cytoplasm starts to break down. So that is called this is the first part of prophase. Now, what will happen in the second, okay, in what we call metaphase. So what happens in metaphase, the chromosomes will actually start to line up. You'll notice that they're lined up on the center of the cell, and each chromosome gets attached to a spindle fiber at its centromere. If you think of a chromosome like an X, right in the center, right there, that is the centromere. Okay, it's the little portion at which holds these 2 V's together, right? And these are basically copies of one another. So what will happen here in mitosis, it'll actually attach to the centromeres, the spindle fibers. You'll see them starting to attach to the centromeres, getting ready to actually pull it apart. Then you come to stage 3. Stage 3 we call anaphase. So we have prophase, metaphase, and anaphase. Anaphase is the point at which the centromeres split. So you notice those spindle fibers had grabbed onto the centromeres, and then it's like tug of war. As they pull, what happens is they end up pulling these chromosomes in half. And so what happens, you have one copy of DNA, these chromosomes here on one side of the cell, another copy over on this side of the cell, and you start to see the spindle fibers drawing away, and the center portion of the cell is starting to thin out. It's getting ready for division. So as the cell stretches out, okay, it pushes things apart and it starts to produce this center portion where it's going to actually divide. Last stage of mitosis is called telophase. And telophase, like telephone, telophase is the point at which the chromosomes actually begin to stretch back out, and they're going to actually ultimately become like the chromatin again. They'll lose that rod-like appearance that they have, and you'll get a new nuclear envelope that forms around the outside. So we now have a nucleus. You'll notice this is starting to stretch out more, getting ready for cell division, and you're actually starting to see real cell nuclei. The centromeres and the spindle fibers start to dissolve and are no longer useful, and that is mitosis. The last stage, okay, is cytokinesis. And let me erase my pen here. Oops, sorry. All right. The last stage is called cytokinesis. This is when ultimately what happens is the cell membrane actually pinches off in the middle of the cell, and you get these 2 daughter cells. So you end up two identical sets of chromosomes, and they have about half the amount of organelles that they needed on each side, and you have the cell cycle. So interphase is number one. Then we have mitosis, which is made up of prophase, metaphase, anaphase, and telophase. And then lastly is cytokinesis, which is the dividing up of the cell. So one thing about cytokinesis that's important for us to understand is that what happens is not only does the cytoplasm divide and the organelles get distributed to each of the daughter cells, but because of the way that it's set up, you get two cells that are pretty similar, pretty close to identical. Now, it happens in two different ways. We can see in this diagram before. In animal cells, the membrane will actually pinch off right here at the center of the cell, and you'll end up getting two cells. You have a cell over here and a cell over here. Those are our daughter cells. In a plant cell, it's a little bit different. A cell plate will form down the center of the cells, and that'll ultimately become the cell wall of each of these plant cells. So you'll end up getting two plant cells that are formed from this one. So a little bit different in cytokinesis between animals and plants, the way that they're formed. There's a great example of the cell cycle activity. If you go to phschool.com, put in the web code CEP 3023, and that's a great little cycle on actually going through the cell cycle. It'll help you understand this a little bit more. Now, one last thing before we move on to DNA. Notice the length of the cycle. People have often wondered, well, how long does this take for a cell to go through one single cycle of cell division? It depends. This is an example over here of a human liver cell. Human liver cell takes about 22 hours to go through the full cycle. The one thing I do want you to notice is the difference in stage one. So stage one would be here, and here, and here. So notice 2 plus 10 plus 19, 21 hours. Okay, is that right? 12, 29, 20, yeah. 21 hours of the time is spent in growth, DNA replication, and preparation for division. That first stage we know as interphase, right? Okay. Notice mitosis is less than 1 hour. Okay. And cytokinesis is, gosh, just a few minutes in the case of a human liver cell. So the process is primarily interphase. Mitosis and cytokinesis are really important too. And this is going to be interesting when you guys do your microscope lab and you look for examples of mitosis. You'll notice that it's hard to find mitosis because of the amount of time that's spent in interphase and not in the other. So pretty interesting little side note. All right, second objective we had was to actually look at DNA and talk about how the structure of DNA is and how that leads to replication in DNA. So want to explain to you the DNA molecule, which is a nucleic acid molecule, is basically supported by proteins and is made up into a shape known as a double helix. It looks like a ladder that's been twisted around itself, and that was discovered by scientists in the 1950s. Crick and Watson came up with this discovery of DNA. Now, DNA can be made up of basically three things, okay? Now, we can see them here from this diagram. They're made up of nitrogen base, okay, a sugar, and a phosphate. So if you listed those things, it's made of a base, which is a nitrogen base, a sugar, which in this case is deoxyribose, and a phosphate. These are the three parts of DNA, okay? The bases are broken down into four types. You have thymine, guanine, cytosine, and adenine. Okay, also known as thymine, guanine, cytosine, and adenine. Either pronunciation is fine. Now, how the ladder's set up, it's really cool, is they're basically replicated. Okay, thymine only connects with adenine. Guanine only connects with cytosine. They're like a key, you can notice in this image here, where they only match. Okay, they use this example. They only match each other. So only guanine can connect to a cytosine on the DNA, and only thymine can connect to adenine. So that's how they're structured. Now, why is that important? Well, let's look at DNA replication. DNA replication is a process by which a DNA molecule actually unwinds, so that ladder that's twisted will start to unwind and unzip and pull apart. and it'll separate into pairs. So you end up having, okay, as this DNA unwinds, you end up having what looks like two rungs of a ladder. Right? So like rung A and rung B. And what's happened is it's unzipped. So this used to be connected, and now it's actually went through this process of unzipping. So what you get is a free half ladder of DNA, and then floating around in the cytoplasm of the cell will be, okay, the nucleus, you'll actually have free nitrogen bases. So maybe you have a guanine and a cytosine and a thymine and an adenine. They're floating around. Now, if you remember, we said adenine only connects to thymine. Guanine only connects to cytosine. Okay, remember those two things we talked about? So, here's what's so cool about this. So how it's been designed, let's say that this is a free ladder of DNA, and I'm just going to give you some little connectors. So let's say this is adenine, guanine, cytosine, thymine. Now, if there is a free thymine floating around, well, thymine only connects to adenine. So this thymine will actually come over and it will connect right here. There's my thymine. And then maybe there's a free cytosine, right? So there's my free cytosine. It's going to come over and it's going to connect with guanine right here. And maybe there's another free cytosine. It's going to connect right here with the other guanine. And there's a free guanine. It'll connect right here with the guanine. And there's a free adenine, and that will connect with the thymine. And so what ends up happening is you end up getting a complete replica of the original DNA molecule, because the way that God's designed it, only thymine connects with adenine, so only free adenine will hook up to thymine. It basically allows DNA to quickly be transcribed and to have a copy of DNA, just because of knowing, if I know just one half, then I know the rest of the DNA just based upon the way that it connects. So super cool how God designed nucleotides, which are the nitrogen bases in the nucleus, to connect with free nucleotides in the cytoplasm or in the nucleus. And one last thing is just to understand that because of the way in which the nitrogen bases pair with one another, the order of those DNA molecules will always match the original DNA molecule. So you can see here's our original, here's our original right here. Notice as it divides up, the free nucleotides come in and connect here, and what you'll end up getting is two of these, right? 1, 2, that are going to be copies of the original. So you'll actually have two copies. And notice how in this example we have the old strand, the new strand, and you can see that these actually become two copies of the original DNA, which is awesome. So that's what happens in DNA replication, and that's how we make new copies, and that's how God designed it. So awesome, really cool thing to know about. So that's WSQ 13. I know that one was a little bit longer, but a lot of really important information. I hope you feel more confident about that. We'll discuss it more, work on that more in class, and you'll have some time to study that a little bit more. We'll hope to see you guys later in class. Make sure you finish up your haiku stuff, your summary, and your questions later.